Thank you, Laura, for that introduction. Well, it's my pleasure to have the opportunity to present the Steve and Sylvia Burgess Lecture this year. And I'd also like to start with a word of thanks, if I can use the right. So I asked Steve's colleagues to provide a few pictures. And so here's a touch of history from Steve as a lad in Australia with his brother, and the early years at the University of Washington, a young professor beginning a stellar career in civil and environmental engineering. And one of the things about these professional journeys that isn't often emphasized, they're shared enterprises, that it's never a journey you take alone, but it's a journey with students, colleagues, and importantly, family. And the department is really appreciative of the Burgess family's very generous support over the years. Steve and Sylvia, thanks so much. So here we go, nitrogen. So I've drawn this presentation from a report that came out several years ago from the National Academy of Engineering, in which the Academy defined 14 grand challenges in engineering for the 21st century. And these are very diverse challenges. I've listed them here, from reverse engineering the brain to fusion energy, but a few of them actually touch upon civil and environmental engineering. So improving the urban infrastructure, providing access to clean water, and challenge number 12, manage the nitrogen cycle. And that'll be the focus of the presentation today. So from the report, they make the point that it doesn't offer as catchy a label as global warming, but human-induced changes in the global nitrogen cycle pose engineering challenges just as critical as coping with the environmental consequences of burning fossil fuels for energy. And that'll be the motif or the motivation for much of what I'll be talking about today, developing a better understanding of this critical nutrient cycle. The other important point, Laura already emphasized that I'm a microbiologist by background, but I've found a real home in environmental engineering over the years, so it's really been a wonderful environment for a microbiologist to develop a career. In fact, I've been in engineering departments much longer than I was ever in a microbiology department. So this is going to be about microbiology as well. So as a refresher course, for those of you that don't think about the global nitrogen cycle on a daily basis, let's get started. Nitrogen in our biosphere is partitioned between three major compartments. The largest reservoir is the atmosphere. Molecular nitrogen makes up, of course, 80% of the atmosphere and the largest reservoir of relatively inert nitrogen. Reactive nitrogen, which is found in terrestrial and marine systems, is bound up in organic matter. Some of the nitrogen is mineralized and bioavailable. But these are not static reservoirs. These are dynamic reservoirs, and there's a very large flux of nitrogen between these different compartments on an annual basis. Now, what drives that flux is microbiology. And when we describe the nitrogen cycle, we usually begin with atmospheric nitrogen, molecular nitrogen, that is brought into the biosphere through nitrogen fixation, a very costly process that some microorganisms mediate. That organic nitrogen, when organisms die, is released through a process of ammonification. That ammonia then has two fates. It can be reassimilated into organic matter, or it can be nitrified. And this will be the main focus of the presentation today, the process of nitrification, where ammonia by a special group of organisms is oxidized first to nitrite and then to nitrate. Now, nitrate is a surrogate for oxygen. Some organisms have the capacity to respire both oxygen and nitrate when oxygen becomes limiting. That nitrate is reduced through a series of steps to nitrous oxide and then ultimately returned to the atmosphere as dinitrogen. So the main points here is that nitrification is essential for completing the global nitrogen cycle, and it links the mineralized and organic forms to denitrification, which returns nitrogen to the atmosphere. If we look more specifically at the process of nitrification, There are some benefits by converting ammonia, which tends to be bound up in the soil matrix, to nitrate. That nitrogen is made more mobile and more available to plants. That's one of the good points, but there are several points that are more responsible for environmental degradation. One is that by making the nitrogen more mobile, it can leach from the environment and then end up in groundwater and other receiving bodies of water, such as lakes and rivers, and of course, ultimately the ocean. And the other problem is this process of denitrification, the respiration of nitrate, reducing it to N2, often stops at N2O. So denitrification generates a significant amount of nitrous oxide. And in addition, the process of nitrification itself releases significant nitrous oxide. This is a very important greenhouse gas, and it's also ozone destructive in the stratosphere. So what has changed was the invention of a chemical process and the industrialization of that process by two Germans, Fritz Haber and Karl Bosch, in the early part of the last century. Now, the Haber-Bosch process, which consumes about 2% of the world's energy supply, it's a very energy-intensive, high-pressure process to bring atmospheric nitrogen into the form of ammonia that is now used primarily for mineral fertilizer. Although at the time it was invented, it was also an important process for the munitions industry. So today, because of this invention, the process, the Haber-Bosch process, this source of nitrogen is a major source of nitrogen for all agriculture. And between 1950 and 2000, the amount of nitrogen fixed to this process and used for agricultural practice increased from about 4 million metric tons now to about 83 just in 2000. So this production of nitrogen fertilizer now supports about half the world's population. It's part of the Green Revolution. And as a consequence, there have been a number of environmental issues that have arisen concurrent with that increased application of nitrogen to the environment. So an important point is that when we apply agricultural application of nitrogen, only about 20% of the nitrogen is actually used by the plant. The remaining nitrogen is lost to the environment either through volatilization as nitrous oxide or ammonia, or lost through leaching primarily as nitrate as a consequence of nitrification. These are both microbial processes. So the loss term in agricultural practice is primarily a microbial process, and management of this loss term will of course depend on better understanding of those important control points, which are microbial. So what are the consequences of this loss of nitrogen from agricultural practice? Here's an extreme example of eutrophication of a lake in China. In the US, we have a major area of oxygen depletion, which we call the dead zone in the Gulf of Mexico. It's a consequence of nutrient transport along the Mississippi River, which drains major agricultural areas in the US. That extends for thousands of miles in the Gulf of Mexico, which has contributed to oxygen-depleted bottom water, and this would not be a place to fish for shrimp. And more locally, nitrogen is very closely monitored with respect to inputs into our local estuary system, Puget Sound. And finally, this also is the primary source of nitrous oxide, an important greenhouse gas, which together with methane contribute to about 23% of total radiative forcing. So they're an important greenhouse gas, important greenhouse gases that are important for ongoing climate modeling, in addition to CO2, of course. So the summary points at this point of the lecture are listed here. So nitrification is essential for the balanced inputs and outputs of reactive nitrogen. We've upset that balance through agricultural application of nitrogen fixed through the Haber-Bosch process, which has led to extensive nitrogen saturation and extensive environmental degradation. And if we're going to support the world population as it grows into the future, mitigation is essential for sustainability. And again, this is almost exclusively a microbial process. Okay. So let's leave Earth for a moment. I'm going to make a point, and I need to take you on a journey. Okay, we're definitely off Earth. So this is the Apollo 11. Neil Armstrong, often quoted line, "That's one small step for a man, one giant leap for mankind." But what is not mentioned, and I don't have any record of this observation, that when Armstrong stepped out on the moon, he did not step out alone. He brought with him a celebratory first step for many other forms of life. This is called the human microbiome. Each one of you carries around with you a burden of microorganisms that is comparable to the number of cells in your body. There are as many microbial cells associated with each one of you as there are cells of your own body. So Armstrong and crew did not travel alone, but carried with them a hidden cargo of billions of microorganisms and thousands of species, many of which are unnamed. So I find this really quite interesting because most of us don't give this much thought. Certainly Neil Armstrong didn't when he stepped out. But why is that so? Why do we have such limited appreciation for microbial life apart from disease and spoilage? Right? So this next few slides will develop the framework for offering a perspective on what we don't know. So this is an adaptation of a pie diagram that was part of the book by E.O. Wilson, The Diversity of Life. I've changed it a little bit. But what Wilson did was try to carve up the described diversity of life as represented in this pie diagram. And there are about 1.4 to 1.5 million described species of life. And what's interesting is most of this is big stuff. Insects, fungi, animals, plants. The little stuff, protozoa, algae, are a much smaller slice of the pie. If you look a little bit more closely at this little red slice, that slice represents described bacterial diversity. As of March 2018, there were only about 16,000 described species of microorganisms. So you might guess that this would be an under-described part of the diversity of life. Right? Doesn't look quite right. So why is that so? One of the reasons is that microbiology is not part of your walk-a-day life. You don't give much thought to the fact that you're carrying around billions of microorganisms with you as you walk through your daily life. So we call them intimate strangers. They are very intimately associated with you and actually determine your general health. And even when we can see them through microscopy, direct microscopic observation wasn't very informative because the simple shapes of bacteria conceal their remarkable diversity. And the other problem is, historically, the only way to describe a new species of microorganism was to isolate that organism in culture, which required that you have the skill to promote growth apart from its normal environmental context. And we now know that most microorganisms don't cooperate. They will not grow in the lab. And many microbiologists have been very frustrated by this, that if you can't replicate their natural niche, they won't cooperate by growing. So what changed? So this was the game changer. The game changer was genome sequencing technology. That offered rather than a culture-based analysis of diversity, a gene-centric census of diversity, which offered, as I'll mention in a few minutes, the opportunity to sidestep cultivation in the description of microbial diversity. And this is a slide, an image of my old thesis advisor, Carl Woese, who really was the forward-looking investigator in bringing gene sequence technology to both evolutionary and phylogenetic analysis of microorganisms. And using a very primitive paper chromatographic sequencing method back in 1975, this is before high-throughput sequencing and human genome, way back in the dark ages of sequencing technology. But even so, the work from his lab offered the first insights into the diversity of life. And now we call this the tree of life, or the big tree sometimes. And what it offers is a true phylogenetic representation of the diversity of all life on this planet. This is based on gene sequence divergence, genomic sequence divergence, so that when we look at this tree, the distance along any segment of the tree is basically a rough estimate of evolutionary distance of evolution through time. And the main observations that came from this first construct of describing the diversity of life was that two of the main lineages of three now that were described are prokaryotic. They're small things that lack a membrane-bound nucleus. This lineage, which represents the eukaryotes, is also mostly microbial. And if we look to these lineages, animals, plants, and fungi, it's actually just a twig on the evolutionary tree of life. And again, to build perspective, this little twig now represents the bulk of described diversity. The red and the yellow, now shown by these two minor segments, not well described. We are missing most of the biological diversity of life in our description of life. So what does that mean? That means that most of microbial diversity, most of biological diversity on this planet remains to be discovered. We call that biological dark matter because these entities are of unknown physiological properties. And there's quite a bit of controversy right now concerning how much remains to be described, and this is based on direct interrogation of environmental samples using DNA sequencing technology. And the argument varies from there are millions of undescribed microbes to there are trillions of undescribed microbes. So the best case scenario is that 99.9% of all microbial species, which represents 99.9% of all biological species on this planet, remain to be formally described. Okay. So the consequences of that is that we have very sparse understanding of the relationships between microbial diversity and the processes that they drive that sustain the biosphere, whether it's in an environmental system or if it's an engineered system that is targeting a specific waste treatment. So where are we at? There are two intertwined problems. How to achieve a sustainable management of nitrogen is needed to support a predicted world population of 9.8 billion by 2050. And also how to advance our sketchy understanding of the microbial drivers of the nitrogen cycle, because the microbes are the important control points for management. So, on to the next half of this lecture. In pursuit of the organisms. What do we know about the drivers of nitrification? Now, before I get into this, this is not really my work. This is the work of many students, postdocs, technicians, and professional colleagues that have worked on this project for well over 20 years. I would love to talk about each of these individuals and the contributions they've made, but I don't have the time to do that. If you recognize people, that's all I'm looking for right now. And since coming to the University of Washington, I've had the great pleasure of working more closely with the departmental members, including Mari Winkler, Stuart Strand, Dave Stencil, and they brought together another collection of really energetic, bright students that are pushing forward the frontiers of nitrogen research. And again, I'll extend my thanks to these people because they are really the story behind the story. And not mentioned here are many undergraduates that have contributed in many ways to the projects as well. So, in the beginning, Russian microbiologist Sergei Winogradsky described nitrifiers in 1890. So, well over a century ago, the first nitrifiers were formally described. And Winogradsky identified two groups of organisms, the ammonia-oxidizing bacteria and the nitrite-oxidizing bacteria we refer to as AOB or NOB, which together oxidize ammonia to nitrate in a two-step process. The first step, oxidation of ammonia to nitrite, and the second step, oxidation of nitrite to nitrate. These are autotrophs. They fix CO2 and grow only on ammonia and minerals. So they're very simple in terms of the nutrients they require for life. More recently, we've discovered organisms, that is the larger community, that have the capacity to combine these two reactions. But for about a century, this was our basic understanding of nitrification. It was a process driven by a few selected groups of bacteria. Now, the story that I'll tell starts over 20 years ago when our lab was involved in a long-term ecological research site in Massachusetts. This is an estuary. And our interest was in how does the diversity and activity of ammonia oxidizing bacteria change along a salinity gradient from a freshwater source to a marine endpoint. And here's one of my postdocs, Ann Bernhard, showing good collection technique. With a hammer, you need to stun the organism before you can begin to culture. But this yielded a surprise. Ann was quite successful in developing enrichment cultures that sustained very high rates of nitrification. But then using all of the molecular tools we had at hand to identify the bacteria that we thought had to be there, she couldn't find them. They weren't there. Good process, but the organisms were absent that we knew should be there. We made similar observations in the study of nitrifying reactor systems in the systems that sustain water quality in marine aquaria, both at the Shed Aquarium in Chicago and the Seattle Aquarium that Laura mentioned a few minutes ago. So we had very active nitrification, but could not identify the bacteria that we knew should be there. So there was obviously something missing, the missing nitrifier that we pursued for several years, and after several years of intensive effort, were able to isolate a novel type of nitrifier. This is the first ammonia oxidizing Archaean, Nitrosopumilus maritimus, the Latin binomial that translates generally into the dwarf nitrifier of the sea. This is isolated from the Seattle Aquarium. It's closely related to Archaea that are highly abundant in the oceans. And it grows pretty much like any other ammonia oxidizing organism by autotrophic growth, that is fixing CO2. At the same time, it oxidizes ammonia to nitrite. The difference, of course, is that this was an organism affiliated with a totally novel group of life. In fact, it defines a new phylum within the Archaea, a major division of life, which are now simply called the AOA, or the ammonia oxidizing Archaea. So having isolated this organism, the next question, well, is it important? And I'll summarize quite a bit of research in just 3 slides. This is a few images from a research cruise I took along with my graduate student Wei Chin back in 2013. We spent a month on the Pacific, the North Pacific, and simply assayed for the presence and activity of ammonia oxidizing Archaea. The general take-home message is that they comprise about 20% of all marine microorganisms. It's a big number. And they are the organisms that control nitrification in the marine environment. When we went to agricultural site, this is work we've done with Stuart Strand, we observed that they also outnumber the bacteria by about 100 to 1. So they're also important in terrestrial systems. And finally, as part of some work we've done in Yellowstone National Park for many years, we also surveyed the park for the presence of these organisms. And surprisingly, we found that these were ubiquitous in hot springs with the capacity to grow up to 80 degrees centigrade. So these are the highest, this is the highest temperature that any nitrifier has. In fact, it's almost 40 degrees higher than the average nitrifier will grow. So we have an incredible range of habitats that these organisms now inhabit. They are the dominant nitrifier on planet Earth. They control nitrification in most terrestrial and marine environments. And 15 years ago, we didn't know they existed. Okay. So why are they so successful, and why did we miss them? So I'll talk a little bit about their novel physiology and their biochemistry, and I'll use that understanding to then address questions about, well, how can we use this new understanding of the diversity of nitrifiers then to improve our application of nitrogen fertilizer in agriculture, and also to possibly improve water treatment technology. So one of the first observations we made that these organisms, these tiny marine organisms, are extremely efficient at eating ammonia. Now, a way to describe efficiency of microbial growth is through a saturation threshold, or a constant known as Km, which simply defines the concentration of nutrient, a limiting nutrient that will support the growth of the organism at half of its maximum growth rate. And for microorganisms, that's how fast they divide. And what we observed is for the ammonia oxidizing Archaea, this half saturation point was down between 10 and 100 nanomoles, as opposed to the bacteria that were about 1,000-fold higher. So they would only start to grow at half their maximum growth rate at a concentration of ammonia 1,000 times higher than the ammonia oxidizing Archaea. So a tremendous variation in their affinity for ammonia. What does that mean? It's hard to really get a sense for what 10 nanomolar or 100 nanomolar ammonia means. But as an example, if you were to take 1 teaspoon of household ammonia and put it into an Olympic-sized swimming pool, that amount of ammonia would support the growth of these organisms at their maximum growth rate. Okay? They would quickly deplete ammonia added to the pool, which is probably added all the time if you have enough kids in there. But in fact, that would be a good place to actually look for these organisms. So the other observation we made was work that we did in collaboration with my previous postdoc, Martin Kunicke, and a biochemist in Germany, Ivan Berg, where we were able to work out a novel pathway for CO2 fixation, which turned out to be a modification of the hydroxypropionate hydroxybutyrate pathway, which probably is not part of your day-to-day vocabulary. But the main point is this is now the most efficient pathway known for CO2 fixation. It requires less energy than the more well-recognized Calvin-Benson pathway, which is used by green plants and also by ammonia oxidizing bacteria. So incredibly high efficiency for ammonia, and now the most efficient pathway for CO2 fixation. So the organism is really designed to compete very effectively with the other groups of ammonia oxidizers represented by the bacteria. So with that background, and there's a lot more that we're not talking about, because physiologically these organisms are really quite unique. I'd like to talk for the remainder of this presentation about present three stories, vignettes that draw from the work I've just described. One will be a marine story, how the isolation of this organism has contributed to developing the solution to a long-standing puzzle, methane production in oxygenated ocean surface waters. We'll talk a little bit about agricultural systems, some work I've done with Stuart Strand on how can we better manage agriculture for reducing leaching and nitrous oxide emissions. And finally, I'll end with some work that really is fairly new, and I'm just a hanger-oner. This is work that's really led by Mari Winkler and Dave Stenzel, but I am really enjoying hanging on to their coattails, so I'll talk a little bit about that. But let's begin with marine systems. So the methane paradox, the marine methane paradox, what is it? Well, back in the latter half of the last century, biological and chemical oceanographers made the observation that there was a source term for methane in the upper 300 meters of the oceans. Okay. If we look now at the predicted concentration of methane if it were in equilibrium with the atmosphere, we see that the upper ocean is supersaturated with a very large source term that drives between 3 and 30% of all global methane emissions. This is a really unconstrained number, 3 to 30%. Why is it a paradox? Well, the only known biological source of methane, and this methane was recognized as a biological source of methane based on isotopic composition, is an obligately anaerobic group of organisms known as methanogens. Methanogen meaning methane generators. Those organisms cannot tolerate a whiff of oxygen, and they're totally absent in the upper water column of the ocean. So what explained this important source term for methane? So for about 50 years, this was really an unresolved mystery. And it turns out it is a phosphorus story, not a carbon story, that ultimately provided the solution to this puzzle. So phosphorus, most phosphorus, as you know, is in a +5 oxidation state, and it's associated with biological molecules through ester bonds. But another form of phosphate is phosphonate, in which rather than an ester bond, the phosphate is tied up with carbon through a direct carbon-phosphorus bond. This is a very stable bond, and there's only one enzyme known that can generally cleave the CP bond, and that's CP lyase, which is encoded by relatively few microorganisms. Phosphonates that you may be more familiar with are phosphomycin, which is a broad-spectrum antibiotic, and glyphosate, aka Roundup, which you also may be familiar with. But in general, we have no idea what the biological function of phosphonates in the marine environment is, even though it makes up a significant fraction of the total organic phosphorus pool. Okay, going on to the solution to the puzzle. What we were able to determine in collaboration with Bill Metcalf and Wilfred Vanderdunk at the University of Illinois, again based on isolation of this organism and genome sequencing, which I haven't described, we identified that this organism should have the capacity to make phosphonate, but we didn't know what the product of that biosynthetic pathway was. But working with Bill, we were able to identify a novel enzyme, which is methylphosphonate synthase, which converts a known intermediate in phosphonate biochemistry, 3-hydroxyethylphosphonate, to methylphosphonate. Okay. So a biological source of methylphosphonate. Well, why is that important? Well, if you remember that enzyme from the previous slide, CP lyase, that enzyme, which is present in quite a few marine organisms, has the capacity to cleave methylphosphate into methane and phosphorus. The phosphorus being used for production and the methane then being released to the atmosphere. So a hypothesis based on the discovery of this novel pathway then was that methylphosphonate generated in the surface waters of the ocean could account for the methane paradox anomaly. And in fact, some recent work out of Dave Carl's group at the University of Hawaii, now examining phosphonate distribution in the upper water column, has in fact verified the production and cleavage of methylphosphonate in the upper 300 meters of the Pacific water column, which could account for virtually all of this anomalous methane production. Okay, so now we have a mechanism for the methane anomaly, the paradox. And having a mechanism, we can now do more appropriate surveys of its distribution, and also knowing the mechanism, we can begin to understand how changes in oceanic chemistry and physics might impact this process as a consequence of climate change. So a relatively important observation, but totally dependent on the isolation of this little organism from the Seattle Aquarium, right? So it's kind of an interesting side story, why the organism is important. So management of nitrification in agricultural practice, and again, the goal here is for improved control of nitrous oxide emissions. And again, this is work that we conducted. It was supported by the Department of Energy. Stuart Strand was a co-investigator, Sally Brown in the College of the Environment, and we had a wonderful colleague at Washington State University, Steve Franson, who was an extension scientist and had been working on test plots of different varieties of switchgrass, okay, which are shown here. Now, as you may know, the Department of Energy invests about almost $500 million a year in their bioenergy research centers. The goal of that investment is to develop a bioenergy economy, converting plants to fuels and value-added products. And one of the plant species that they think might be a good fuel feedstock is switchgrass, one of the last of the tall grass prairie plants, a perennial that has very good yields, relatively low nitrogen requirements. So our interest was, well, under managed conditions of nitrogen application and tillage and these different plantings that I won't describe in detail, what's the impact on nitrification? And the first observation we made was that management totally transformed the nitrifier community, whether it was a tilled field or it was a perennial planting of switchgrass. You can see here that the native, this is a PCA type analysis using molecular markers to relate the native community to the communities that emerged after management. And you can see that there was a whole scale transition in the nitrifier community. And if you look at the phylogenetic relationships, the native genotypes were totally replaced by these managed genotypes. So really quite a profound impact on the native microbial community as a consequence of these switchgrass plantings. And of course then the question arose, well, what is the impact of this total transition in community structure on nitrous oxide production at this site? And we, working with Sally Brown, collected using flux chambers nitrous oxide emission data. And again, to make a very long story, a lot of work reduced to a couple of slides, the observation was that nitrogen addition was correlated with a rapid increase in the abundance of ammonia oxidizing bacteria, and that increase in the ammonia oxidizing bacteria was correlated with high production of nitrous oxide. And there was very good, across a number of study sites, very good correlation between the abundance of the ammonia oxidizing bacteria and nitrous oxide flux. When we then looked at the Archaea, they were listening to a different drummer entirely. In fact, they were just almost totally unresponsive to nitrogen addition. In fact, their numbers seemed to be depressed when fertilizer was added to these switchgrass fields, even though their abundance was about 10-fold higher than the bacteria. Okay, so what's the take-home lesson here? This is a relatively straightforward take-home lesson based on the physiology of these organisms. Now we have an understanding of the relative contributions to ammonia oxidation and to nitrous oxide production. The AOB are what we call copiotrophs, big eaters. Copiotroph, meaning eat big. Grow only at high ammonia concentrations. They grow very fast when ammonia is available, and they produce a lot of nitrous oxide when they're growing. In fact, all of the nitrous oxide produced in these switchgrass fields is derived from ammonia oxidation, not from denitrification. The AOA, in contrast, are oligotrophs, meaning little eaters. They grow at very low ammonia, like a teaspoon in an Olympic-sized swimming pool. They grow very slowly, and they don't produce a lot of nitrous oxide when they grow. So the solution, in large part, to controlling nitrous oxide and nitrate leaching is controlled release of fertilizer. Now, this is not big insight, because we know that split application of fertilizer is advantageous in reducing environmental damage. But now we have a very clear mechanism to control the amount of nitrogen that we want to add to a field through a slow release format, to reduce the AOB contribution, keeping ammonia below their growth threshold, but also promoting the growth of the ammonia oxidizing Archaea, which increase the availability of nitrogen by oxidizing ammonia to nitrite for better delivery to the plant, and at the same time reducing nitrous oxide production. So it would be a win-win if we could control the ammonia that's made available to the nitrifiers in this system. Okay, the last bit of this presentation. This again is work that's mostly led by Dave Stenzel and Mari Winkler, and I am just riding their coattails. So it's a good ride, though. So this is the West Point wastewater treatment plant. Takes up a lot of real estate in a very nice location below Discovery Park. And what Dave and Mari are involved in is developing new technology that will reduce the footprint as well as the energy demand of wastewater treatment facilities such as this. And with their graduate students, and they include Max and Bao and Stephanie and others that I'm probably forgetting to mention, they have developed a pilot system. This is a system that was developed by Dave and Mari, and with the also the contribution of a process engineer from King County, Bob Booker. And this reactor system is designed through operation to select for this type of microbial community. These are small biogranules, about 1 millimeter or 2 in diameter. And these are just really powerful microbial factories. You pack a lot of biochemistry into a very small particle, and by doing that, you can generate a system where you have very high process density, so very high rates of conversion in a very small reactor volume. So the process has been followed for, I don't know, almost 9 months now, I guess, right, Max? Something like that. And this shows the startup of the system and the increase in ammonia and nitrogen removal rates, converting ammonia to dinitrogen gas in these systems. And ultimately, this will be integrated with a mainstream reactor system to also promote more efficient removal of nitrogen from the mainstream reactor as well. But note that it takes quite a while for this reactor to get up to full steam. And there's also been some issues of stability. And these are not always happy systems. They don't like changes in the operating conditions, as Max can testify to. But they are very efficient once you get them running. Now, what Mari Winkler would like to do is to avoid some of that startup hassle, to begin with the community you want rather than select for the community you hope to achieve, right? So the idea is to build simple synthetic microbial communities and encase them in small polymer beads. So in this case, we're going to trap AOA and Anammox microbes in synthetic biogranules. The idea then for this format would be to have Anammox, which is an organism that converts ammonia to dinitrogen using nitrite as an oxidant, with an ammonia oxidizing archaeal outer shell, which would partially oxidize the ammonia and deliver both ammonia and nitrite to the inner core, which is anaerobic, and drive then rapid and efficient conversion of ammonia to dinitrogen gas without any organic addition. Okay, so that concept is now being realized in the lab in these small reactor systems, and these hydrogel biogranules containing ammonia oxidizing archaea and Anammox microbes are being made in almost production scale now by Bruce and Matthew working in Mari's group. And these are now being placed in pilot reactors in the laboratory and beginning to develop the foundation for what we call a synthetic wastewater treatment technology, where we completely control the architecture of the community that's involved in the wastewater treatment. And that can, in principle, also be used for more portable application of wastewater treatment and very small scale operations. The other part of interest is that imagine if you can actually design a biogranule that drives the process of interest. Here we're looking at nitrogen removal, but a big problem that Stuart Strand has worked with for many years are pharmaceutical byproducts, estrogenic compounds that pass through current wastewater treatment plants unchanged. They often are estrogenic, they cause feminization of fish, they have other adverse effects on our natural biosystems. Imagine if you could design a biogranule, Stuart, that would selectively target estrogenic compounds. That would be a wonderful advance in wastewater treatment technology. So I will stop at that point. And there's much more to tell. That'll have to wait until another time. And what's the main take-home lesson is that I hope you have an appreciation for the organism, that understanding the organism offers important insights that have very practical application. And thank you for your time.